Well, I'd like to welcome Joe Glorioso from the University of Pittsburgh. Joe will be giving us a little bit of a different take, talking about the translational application of HSV gene vectors in the context of nervous system diseases. Thank you, Joe.
I'm sorry I'm not talking about oncolytic viruses, but I was asked to talk about developing herpes vectors for treating neurologic diseases, and today I'm going to concentrate on treatments for chronic pain. I have two disclosures. I was a co-founder of several startup biotech companies, Oncourse Inc., which is an oncolytic virus company in Cambridge, and Coda Biotherapeutics, which is a pain gene therapy company in San Francisco. So how do we recognize that we have pain? So what happens is that there are cells in the peripheral nervous systems, and these sensory neurons, once they're activated, will release, pushed the wrong button, sorry. So they will release neurotransmitters in the dorsal horn of the spinal cord, and this will activate second-order neurons, which will deliver signal up the spinal thalamic tract, where third-order neurons are activated in the cortex, and this is how you become aware that you have pain. So it turns out that there are several kinds of fibers that will deliver pain signals. These are nociceptors, and one type is called A-beta fibers. A-delta fibers, sorry. And these fibers are myelinated, and they're large diameter fibers, and they deliver signals very quickly. So what you'll see is that these will lead to acute pain recognition. This is, of course, protective. The C fibers are not myelinated. They're small diameter nerves, and they will deliver signals more slowly, and this will lead to chronic pain, which could be burning or a deep aching pain. So what is the problem here? Why do we have chronic pain? And what really happens is that there is a change in the biology of these neurons so that they will become hypersensitive to normal signals. So for example, if you look down here, you'll get hyperalgesia or allodynia as a result of changing the threshold in which these pain fibers will react. So why do we need gene therapy for pain? It's because the drugs which are used are given systemically, and the systemic use of these drugs can lead to a lot of unwanted side effects. For example, everybody's aware of the oxycodone crisis that's going on right now. So there really is no long-term drug therapy for chronic pain. So for example, then we're looking for vectors that will be effective in treatment of this kind of pain, and herpes simplex is a logical one because it establishes latency in C fibers where chronic pain arises. These vectors can be rendered safe, non-replicating, and non-toxic and non-inflammatory. They can persist in ganglia as non-integrating circular episomes and act as a platform for expressing therapeutic agents. They're high capacity, so you can put large or multiple gene cassettes into them, and you'll be treating pain where pain arises, and that's very important. These vectors can also be targeted by infecting the dermatome, and depending upon the radicular distribution of these nociceptors, you can infect the specific ganglia. And you can also, there's a number of other ways that you can target these, the expression of these products. So it turns out that over the years we've tried a number of ways to treat chronic pain, and these include methods for silencing pain signaling from sensory neurons and producing products like enkephalin, endomorphin, and GABA. We block conditions that induce hypersensitivity. For example, we can alter ion channel functions or signaling pathways. or we can antagonize inflammatory cytokines. We've done all of these things over the years. And eventually, if we understood better how chronic pain neurons the pathology has changed, that we may be able to actually correct this. So how do we develop these vectors? I'm going to talk about herpes simplex. This is a virus which expresses its genes in waves called cascade regulation. And what happens is there's a protein in the virus particle itself that binds to the immediate early promoters and will activate expression of these immediate early genes. These will go on to make early genes which are involved in DNA replication. And finally, after replication, you get these late genes in virus assembly. So the idea is then to knock out or render inactive all of these immediate early genes, and this blocks the whole virus cascade. These kinds of viruses then don't express any lytic functions. There's loss of toxicity, and these viruses then cannot replicate on their own. You have to engineer cell lines which will complement these defective genes and trans in order to make them. We've also deleted the joint sequence of this virus, which is about 14 kilobases, and this provides room for putting very large expression cassettes in the virus, and then we've been able to figure out how to express transgenes long term. So one of the problems with these viruses is that when you remove ICP0, this protein, this immediate early protein, is very important in keeping the virus genome transcriptionally active. So how do we solve this problem? We worked on this for a very long time. It turns out that that David Bloom a number of years ago demonstrated that there are these insulator boundary elements that are in the genome strategically placed around the latency gene, which is active in the nervous system, and some of these other immediate early genes. And what he's shown recently is that a specific transcription factor called CTCF will bind to these and will prevent reactivation of virus from latency. But so what we did was then we positioned the expression cassettes in between these insulator boundary elements, and what we found is that we could get long-term transgene expression as a result. And you can see that these boundary elements then can block the spread of methylation in the sequences that you want to express, and they can also form loop outs of the DNA by homodimerization. So this is a vector created several years ago by Yoshi Yamanaka, and what he did is to introduce a gene cassette into the latency region flanked by these CTRL sequences, and also we introduced a reporter gene, M. cherry, under ubiquitin control, and we did this really just to watch the virus for growth. We had no idea it was going to express in the nervous system. So what we found was that you can see here that you get very good expression. Here's out of 30 days, and we've seen expression for many months, as long as we've looked from these gene cassettes. And it turns out that that this particular construct is expressed in the central nervous system in neurons, but this one is not, and that's a surprise. And in fact, this one expresses in fibroblasts, which are not part of the nervous system. So we're currently trying to figure out how this is working, but this is a beautiful way to express gene cassettes long-term in neurons. So I'm going to talk a little bit about pain now. There are two kinds of conditions that we're going to discuss. One is chronic bladder pain, or interstitial cystitis, a model for this, and neurogenic bladder pain. So the early studies we did were to use enkephalin, and this is an HSV vector that we express this natural opiate peptide, and it binds to receptors which are similar to what these opiates that people give to treat chronic pain. So the technique is to inject the virus into the skin, or in this case into the bladder wall and the neck of the bladder. The virus is taken up by these nerve terminals, the C fibers and A delta fibers. The virus is retrogradally transported to the nerve cell body, and then after this, this afferent starts to express enkephalin, and expressing enkephalin locally in the nociceptor afferent causes the release of enkephalin. It binds back to its receptors, delta mu receptors, and these will block calcium-dependent release of these neurotransmitters. And in addition, enkephalin will block these second-order neurons from functioning. So this is a potent way to block the pain response. So in the bladder, what we've done is inject the virus in the bladder wall. the virus is taken up by these ganglia. Sorry, sorry about that. Sorry, L6 and S1, L1 as well. And once the virus is into the ganglia, it will start to produce enkephalin. It'll be released in the spinal cord and block pain transmission. In addition, it'll block neuropeptide production and cytokine release at the bladder wall. Now, this doesn't affect the sphincter, bladder sphincter, which is controlled by the peduntal nerve. And what happens here is that you'll get different kinds of pain demonstration. These animals will lick, the genitals and not much freezing, whereas in the case of the pelvic nerve, you get a lot of freezing and very little licking. So this is the experimental methodology. The virus is injected, either a control virus or the enkephalin-producing virus, into the bladder wall. Wait a couple of weeks to allow good expression. Then we introduce saline into the bladder, and after about 15 minutes, we inject either capsaicin or racinoferatoxin, which causes a burning pain due to the activation of the TRPV1 receptor. And then if you look at the behavior of these animals over this time and voiding behavior, you can determine to what extent these enkephalin is having an effect on bladder pain. So if you look at what happens with the enkephalin vector, here's the saline introduction and injection of racinoferatoxin. This is quite a different result. You don't see these interstitial contractions that like you see in the case where you have the control vector. You can reverse this with naloxone H, which is a form of naloxone that goes into the spinal cord, whereas naloxone M does not, because it doesn't reach the right nerve terminals. So I think this is a dramatic demonstration of how enkephalin can play an important role in controlling this kind of pain. And if you look at the behavior of these animals, the animals that were injected with the enkephalin vector are showing a dramatic difference in pain-related behaviors. You can see what it looks like, and much less so for this type of behavior, because this is driven by a different part of the bladder. Now, there's some limitations with using continuous gene therapy with a gene like enkephalin, and that is that we purposely did the first clinical trials here by using a promoter that would be short-term, several weeks. So we don't know what would happen if you express enkephalin long-term. Will there be tolerance or other unwanted side effects? And in addition, you can't turn this off when it's no longer needed. So the solution, we think, is to deliver a drug-regulated gene product, that can block nociception signaling, allowing a kind of on-off switch without compromising nerve function. So to do this, we turn to the glycine receptor, and this is a ligand-gated ion channel that will channel glycine into the cell, not glycine but chloride rather, sorry, into the cell, and will cause hyperpolarization. This is present in the spinal cord and the brain as well, but not in peripheral nerves. So this is kind of ideal, so you can introduce that gene into a specific set of neurons, which is, and these are the only ones that are expressed in the peripheral nervous system. So the idea is to take a virus which mediated the wild-type glycine receptor and look at pain signaling after the chloride is channeled into the cell. And we only use the alpha subunit of the glycine receptor, because if you use both subunits, it distributes the glycine receptor differently. With the glycine receptor alpha alone, you can get up and down the axon, which is ideal. So this is a similar model where we injected the vector, control vector, and the glycine receptor vector. And what you can see here is that when you add the drug, in this case it's just simply glycine, we just injected it IP, with a lower concentration and a higher concentration, it had no effect on these rapid bladder contractions. On the other hand, when the animals receive the glycine receptor and you add the drug, which is in this case just glycine, when you get up to 1 milligram per kg, you get a dramatic effect. That is, the glycine channel opens up and now it blocks this painful stimulus. The problem here is that the glycine receptor is also in the central nervous system, and so what we had to do was figure out a way that we can improve the targeting of this drug. So several years ago, Joe Lynch from Australia published a nice paper to show that, published a nice paper to show that that a double mutation in the glycine receptor would make it insensitive to glycine. But of interest was the fact that this double mutation caused a dramatic increase in activation by an anti-Helminth drug called ivermectin. So to show that we could differentially activate these two receptors, we used what's called a paw withdrawal latency test. We put the animal in a Hargrave apparatus, which is a cage in which there's a glass floor that you shine a light bulb onto it and raises the temperature up to 40 degrees. And analgesia is measured by how fast he'll raise his foot. So the animal that got the normal glycine receptor is responsive to glycine, is an analgesic response. But the animal that got the ivermectin responsive channel was not responsive to glycine, but was responsive to ivermectin. So what we have here is a way to target activation of this receptor using a drug that was only reactive with the receptor that was put into the peripheral nervous system. So I'm going to talk now about neurogenic bladder, which is a problem in spinal cord injury patients. There are many of these in the United States. They occur every year. They're mostly in young people. And all of them experience eventually bladder dysfunction. And what this means is that they have chronic leakage of the bladder. And this incontinence causes them to have to catheterize many, many times more than normal. And this eventually can lead to urinary tract infections, which can be quite serious. So this is what happens in the normal types of voiding that babies, when they're born, have incontinence. As they mature, they develop voluntary control, which is mediated by the brain. And this is lost over time, which if you have some of these diseases or in case of spinal cord injury. This leads to an involuntary pattern, which is something similar to infants. And the idea of therapy is to reverse this back to a voluntary system. So most of these injuries occur in this part of the spine, or maybe a little higher. And there are a couple of things that happen. First of all, there's a loss of bladder filling, and then there's a problem with the connection between the opening and closing of the sphincter with bladder contractions. So these patients never can fully empty the bladder. And when the bladder is contracting, the sphincter may not be open, and vice versa. So there's a loss of this kind of control. Okay, so here's what happens at a cellular level, that there is a change in the biology of these C fibers, which will drive this pathway in the spinal cord, and this will end up pushing these other neurons, which is the efferents, to open and close the sphincter, bladder sphincter. And what we wanted to do here is see if we could target expression of a transgene, in this case it's just M-cherry, in these particular neurons, and ask, what sort of phenotype do they have? And we used a promoter that was active in TRPV1 positive cells, these peptidergic neurons, CGRP, as well as NF200, which are not infected generally. And we used this more non-specific promoter, CMV. Okay, so in this experiment, what we're showing here is that that after you inject the virus, we would look at the ganglia, and there are two ganglia we did in these experiments, L1 and L6. They're both infected by the virus, and you can see with the CMV promoter-driven M-cherry, there was no change in the numbers of neurons. This is pre-spinal cord injury. This is post. And so CMV is expressing in both these kinds of cells, but you don't get an increase in expression. However, what we found was that in CGRP positive cells, that M-cherry was upregulated. We found more examples of M-cherry positive cells in injury patients, both in these two ganglia. So this is telling us that the nature of these cells are changing, and this is part of the phenotype. In addition, if you look at the NF200 promoter, it's not active in cells because these are cells that are mostly not infected by the virus. And finally, we looked at TRPV1 positive cells, and there was a dramatic increase in the number of cells that were expressing TRPV1, and we could tell those cells by using the TRPV1 promoter driving M-cherry. So what we're seeing here is a change in the phenotype of these cells in a way that explains why there's hypersensitivity. So this is the last experiment. I'll show you that what we did now was to take the ivermectin sensitive glycine channel, which is a mutant glycine channel, and we introduced it into the bladder wall, as I showed you in the past. And you give ivermectin the drug, and you have the normal with a normal receptor, you don't see any decrease in the number of these spasms that occur in the bladder, and there's incomplete draining of the bladder. So, however, with the virus where it's responsive to ivermectin, you see a dramatic shift in the number of these bladder contractions, and there's also a change in the micturition pattern. So this shows you that ivermectin has a dramatic effect on these bladder contractions, and you get a dramatic effect on complete urination. So this is the purpose of this gene therapy. So to conclude, I'll just say that HSV is are vectors that are at hand for safe long-term transgene expression in sensory nerves. Gene therapy for bladder pain and neurogenic bladder is feasible. The drug, we can use drug-regulated nociceptor silencing as an important advance, and we can target specific subpopulations of these nociceptors with these ligand-gated chloride channels. So now where we are is we want to use these specific promoters to express the ivermectin-sensitive channel in subpopulations of neurons, specifically C fibers, that cause this hyperactivity in the bladder. And so this is going to amount to building more viruses and testing, but I think we're poised to do some really interesting final tests in this. And by the way, the drug here is given systemically, and that's an important feature here because we need to provide these drugs either by mouth or locally in a cream or something, so that we can activate these responsive channels. So this work was done in two different departments. Naoki Yoshimura and several of his postdocs did the experiments that I'll show you most of the time. This is the targeting experiments to the nociceptors in the ganglia in the bladder experiments. Dr. Yokoyama was involved in doing the work with the enkephalin response. And in my lab, and along with Bill Goins, we've done the rest of the work looking at the glycine receptor with using glycine. Yoshi Miyagawa was the person that developed all these interesting vectors that we've used. Marco Mazzulli was the person that created the transgene viruses, and Mark Doyle was involved in the targeting experiments. Thank you.
This paper is now open.
Joe, I got a first question. So if you were to do a trial for pain control where you inject HSV with a lot of human experience, so it's quite safe, how do you dose escalate? Is it, like try to get it to as early as possible with your first dose level to a meaningful dose that you produce enough protein for control? Can you kind of like what would be the ideal clinical trial that you do so that you
Yeah.
have minimum exposure at subtherapeutic levels?
Well, you can do dosing experiments. We've done those in animals, but there's not enough experience in humans to know. But for these viruses, when you inject into the skin and they're taken up by nerve terminals and retrogradely transported to the nerve cell body, it's a very efficient process. So in animal studies, we've used about 10 to the 7th PFU, as opposed to some of the vectors that people are familiar with where they use enormously higher numbers. When we did a patient trial some years ago for pain related to cancer, we didn't see evidence of inflammation, and there was a dose response, although there was this was a low number of patients. So we haven't repeated that.
Other questions? No? Thank you very much.
Thank you.